Welcome to the Watchlist. Joining us is Panna Sharma, who is the CEO of Lantern Pharma. Panna and his team have been busy working on some fascinating drug developments. So hello and welcome. So happy to talk to you today.
Right, thanks for having me on. I appreciate it.
Jumping right into it. So how did the LP-184 trial stack up against others aimed at the same cancers or DNA damage targets?
Yeah, it was a fairly unique molecule. One of the things that we thought about LP-184 when we were developing is the molecule we believed would be more potent than traditional DNA damaging agents, which it seems to be. But also, unlike a lot of these highly potent agents, it's really activated at the site of the cancer cell. And so that's what makes it somewhat unusual or unique, is that the drug actually doesn't become a killing agent until it's inside the cancer cell. And it does that because it's been designed to get activated by the presence of a specific enzyme, PTGR1. And when that's present, the molecule lights up. And so we found that to be the case. So although the therapeutic window is very workable, we didn't really have any significant adverse events that were not manageable. And the dose limiting toxicity window was really, really high for the drug. So we know that the drug has a very favorable tolerability and safety profile, one that can be managed as monotherapy, and one that can also be exploited in combination therapy. And so that's typically pretty unusual for DNA damaging agents, is when they are DNA damaging at that level of potency, it's hard to combine them with other drugs because you get a runaway toxicity profile both inside and outside the tumor. But that's not the case here. So this gives us a lot of excitement that this drug will work really well in a wide range of cancers, both as monotherapy, but also potentially in combination therapy, which gives it a much more robust profile in cancer.
And are you able to speak to plans in place for testing LP-184 with PARP inhibitors or immunotherapies? And would you be able to share which combination would reach patients first?
Sure. We've been publishing on the combination potential for LP-184 for quite a number of years when we first started discovering that it was really synergistic with other agents. We theorized that it would work really well with PARP inhibitors. And when we saw that come to life in our own laboratory studies, but then also in independent laboratory studies that we did with Dana-Farber, with Danish Cancer Research Institute, most recently with UMass Boston, every one of those researchers found that our drug, when combined with PARP, had a pretty dramatic impact across these cancers. And the mechanistic rationale is actually quite interesting. So I'm going to touch on that. Our drug works by destroying the DNA of a cancer cell, double-stranded breaks. So it rips them apart at a very unique site, very different position. And then what happens with PARP inhibitors work almost on the opposite end of the spectrum. They work by stopping or halting the cancer cell's ability to repair itself. So we're working on two very different sides of the equation. We're damaging it through our drug, and PARP inhibitors are blocking the cell from repairing itself. So it's really kind of a combination 1-2 punch that's perfect. No real overlapping mechanistic rationale, but it creates this massive synergy. And that's what is actually very unique also about the drug, is that we're hitting both bookends of both destroying and stopping the repair. And so what we've seen in triple negative breast cancer is, regardless if the triple negative breast cancer is actually sensitive to PARP or not, it becomes sensitive to PARP. And so in all the triple negative breast cancer cell lines that we've tested on our own, but also through our collaborators, we found that across the full range of TNBC cell lines, it's been able to regress the tumors by 100%, which is pretty drastic.
Definitely sounds like there's lots coming down the pipe. And I know you're a busy man, so I'll keep it short with the last question. But how does the AI platform RADR help make the drug trials potentially faster or cheaper or more likely to succeed?
Yeah, so a lot of the work that I talked about in terms of determining the mechanistic rationales all was done largely through the help of AI. We saved years and years of work by doing that. The idea of TNBC from scratch to now, I mean, that could be, half a decade, and that took us less than probably a year and a half. Now, going into the clinic, coming up with a biomarker selection process will also make our trials more streamlined, save time and money, and also give us insight as to why some patients respond and others don't. So, AI and, in fact, data-driven processes, I think, are going to be central in all future oncology trials. And we're at the forefront of that process. And our entire business model is built around a data-driven AI-first approach to cancer drug development, both preclinically and also in the clinical setting.
Yeah, it does sound like AI is going to be the future or is the future of your field.
It's now.
Well, thanks for your update, and Panna, may the clinical trials be nothing but good news from here on out.
I hope so too as well. Thank you for your time.
Thanks. Again, in case you missed it, that was Panna Sharma, who is the CEO of Lantern Pharma. Thanks for watching the watch list, and I'm Brianna McCutcheon.